Hello, welcome to our STEM News broadcast for week 2 of winter quarter of 2022. We are STEMNewsNexus at UCLA, blog of preprints and new publications. A little bit more about us is that we are an undergraduate student organization at UCLA, and we are just really passionate about research. We're really just trying to squeeze in that extra article published within the last 4 weeks that has nothing to do with our exams, research, or fellowship applications. We are actively looking for faculty members to help us interpret the data and learn about different lab techniques. So if you're interested, please email us at STEMNewsNexus@ucla.edu. And if you're an undergraduate that's interested in joining us, you can find our application on our linktree/STEMNewsNexus. So the purpose of our organization is to make these scientific discoveries more accessible. As mentioned, we are constantly reading papers for something, an exam or a grant or a research project, but we're never just looking at the plethora of science that's just growing each day. So we want, and just like, it's kind of difficult to read those articles because they're so dense. So we think that by splitting up the task by reading different papers and creating these videos, we can help make it more accessible for all scientists and developing scientists in our community. And so the structure of this is that we have different slides summarizing our blog posts that we will publish on STEMNewsNexus.com, but our release date is to be announced. So let's get started with some STEM news.
Hi, everyone. My name is Maria. And today I'm going to be talking about COVID-19 and its prevalence in older populations. The study that I read was Age-Associated SARS-CoV-2 Breakthrough Infection and Changes in Immune Response in Mouse Models. What this article showed was that older mice showed increased susceptibility to reinfection due to insufficient immune protection with the first infection. What was really interesting about this study was that 2 doses of the COVID-19 mRNA vaccine in older populations showed limited immune responses, making them susceptible to reinfection. The older mouse in this study displayed more severe symptoms, which included tissue damage and severe inflammation, consisting of congestion, edema, and localized hemorrhage. An interesting piece of evidence that I found in this article is as follows. While infected young mice did not show any sign of disease, the aged mice showed ruffled fur, hunched postures, and labored breathing. I think that this is really similar to what we saw happening in the nursing homes and in the retirement communities earlier in the pandemic. We saw that the older population displayed more severe symptoms and in general had a larger struggle of working through the COVID-19 pandemic and the virus itself. Thank you for listening.
The official title of the work is A Naturally Inspired Antibiotic to Target Multi-Drug Resistant Pathogens, and it's by Zhongcheng Wang et al., published on January 5th of 2022. And they are focusing on the antibiotic colistin, that is the last known line of defense against gram-negative pathogens. And it's becoming less effective with these pathogens that carry the MCR1 gene. So the scientists found a structurally divergent colistin congener called maclolacin, and they proved that maclolacin is effective against MCR1 gram-negative pathogens.
Hi, everyone. My name is Lawrence, and today I'm going to be talking to you about a new discovery regarding a new layer of the jaw muscle. Now, this jaw muscle in question is called the masseter muscle, and it is located at the back of the cheeks. So when you bite down, you can feel it contract, and when you relax the jaw, you can also feel the muscles relax. So if you put your fingers over here and you bite down, you should feel it contract. Now, prior to this new discovery, the masseter muscle is commonly described as having 2 layers, a superficial layer, which is the surface layer, and then you also have a deep layer, which is the deeper layer. Now, previously, there are also claims that there is a 3rd layer present. However, a lot of those claims are contradictory. Therefore, This study aims to comprehensively examine the structure of the masseter muscle so that the strength of the claims of a third layer being present can be determined. Now, the method being used was that cadavers were obtained, and then the head were analyzed using methods such as dissections. Now, these methods allowed for the exposure of a new layer, which is called the coronoid article. Now, it is important to note that in all of the cadavers studied, the coronoid layer was present. This means that the presence of this coronoid layer is not an anatomical variation. Rather, it is a constant presence. Now, the significance of this layer is that this layer is distinct in terms of both its orientation and measurement. Therefore, this is evidence that it provides further functional stabilization of the lower jaw, just like the masseter muscle does as a whole.
Human immunodeficiency virus reduction with natural killer cell therapy enhanced with SUW133 protein. The official title of the work is Latency Reversal plus Natural Killer Cells Diminish HIV Reservoir in Vivo by Jocelyn T. Kim et al., published on January 10th, 2022. So essentially, these researchers took human natural killer cells and CD4 plus cells. They infected the CD4 plus cells with HIV and then saw if the natural killer cells were activated in vitro in the presence of the CD4 plus cells, the infected ones, and they did. Then they decided to proceed with the study, and they took these human natural killer cells and injected them into mice after mice that were kind of modified to have HIV. They were genetically modified mice, and they also had tissue implanted into them that contained HIV within it. And they were also infected with HIV. So, but what they did, and the HIV that was made had a special barcode that was like a fingerprint. And we'll talk about the transfection in a little bit. But basically what happens is normally humans have these antiretroviral therapies that try to keep the amount of HIV in their body low. And eventually there are breaks in taking these medications. So then the HIV virus then repopulates in the blood, killing and then causing like all the symptoms of AIDS. So then, but they kind of just wait until the art is working to lower all the HIV in the mice, and then they stop it, and then they want to check how fast the amount of HIV repopulates the blood. And they do this under two conditions. So in one group is just the experimental control, and the other one is treated with natural killer cells. So these are cells that can find infected cells in the immune system and then destroy them before they can go and infect other cells. So then this can prevent like replication or reduce the amount of virus factories or destroy virus factories before more viruses are made. So what they noticed is that the mice that were treated with the natural killer cells that were like destroying all the virus factories allegedly did have a reduced rate in viremia within a week when they were constantly measuring the blood of the mice and then checking and quantifying how many of those viral signature barcodes are in the blood. And they noticed that the viral barcodes appeared much faster in the control group than in the experimental group, but eventually they both had HIV markers after 2 weeks of art discontinuation. So, and they also then dissected them after death in the necroscopy, and they noticed that the amount of HIV in the mice treated with natural killer cells was significantly lower than in the control. They also noticed that the mice that were treated with the natural killer cells just had more immune cells or more CD4 plus cells in their body. And they also noticed that they double checked that there was no difference between the infection and cell implantation into the mice, which means that the differences that we just discussed are not because of systemic error where they kind of just put less HIV in the experimental group. They put the same, and the experimental group was only different because of the treatment. And Then they quantified the rate and noticed that it was in the experimental group and noticed that the relapse was 2.1 times slower. And they also noticed that the viral genomes that they were collecting from the mice had way less mutations in the barcodes, meaning that there were less strain generation HIV, which is one of the things that makes HIV really difficult to cure. But then later, once earlier I mentioned that these mice were modified with tissue implantation. So they took the tissue explants and then they were from both groups and they were able to then induce them out of latency and get them to generate more HIV and in vitro. So this suggests that although the natural killer cell treatment was somehow killing cells, killing cells in the body that were infected with HIV, they were not targeting the latent ones that were implanted, but were just targeting the ones that were recently infected and active and generating or being like active viral factories. And so from literature reviews or other studies, they've noticed that UW133 can make a cell come out of latency and become active again. So they wanted to see if it was actually the latency thing that made the natural killer cells unable to detect the reservoir of HIV. And so they decided to undo the latency and make those like initially latent cells active. Then they noted, and then by administering this like active viral factory inducer, SUW133, in conjunction with natural killer cells, they were able to get the lowest rates of viremia, almost no viremia, and they were able to then also have highest rates of immune cells within the mice, and also had the least amount of clones in the viral clones that existed in the body. So this basically suggested that they can use natural killer cell therapy in conjunction with cells that undo latency, so that the natural killer cells can better identify HIV, host cells containing HIV, and kill them before they can generate a lot of HIV in the body, to eventually be like some sort of cure some sort of cure for HIV, possibly. So the techniques, as mentioned, they did transfection. So this means that they have this plasmid that has all the proteins for HIV, but also has a barcode. And then they put it on top of these 293 T cells, which uptake it. And I think if this plasmid is similar to Fugene, then from what I interpreted, the methods, which were kind of brief, that it would integrate into the host genome and then hijack the cell factory to generate a lot of HIV cells, and also that they use specifically 293 T cells because their membranes, once the exocytosis is occurring with all of the viral particles, that those membranes are stable enough to generate a lot of viruses in the supernatant. So once they have the viruses in the supernatant, then they can use those to infect CD4 cells. Then they also did centrifugation. So when they're collecting blood from the mice or from humans, they separate the white blood cells from the red blood cells. And so then they're able to get the natural killer cells from the white blood cells. They also did FACS sorting, which is like they add different antibodies to bind to different proteins on the surface of the cell, and that these antibodies have a protein on them that when it absorbs fluorescent light, it'll emit light in a lower wavelength, but that those light emissions can be detected. And when there's a presence of the light, that means that the antigen surface marker exists because the antibody could bind to it. So they did this to identify the natural killer cells with cell surface markers. They also then did the barcode analysis. So once they took all the RNA from the blood, they were able to amplify it and then sequence it with Illumina to identify and quantify the viral barcodes. And they confirmed that there was no biasing by repeating it twice and seeing that there was no difference between the sampling size in both attempts. And Yes. And so I think the strong point in the paper is that they kind of were able to take the do the explantation experiment and show that the natural killer cells were not affecting latent cells, because they showed that they can make those explants generate viruses, but then they were able to basically then they knew that they were latent, and so they knew that adding SUW133 would make it not latent, and then they were able to get better results later in the study. So this kind of just shows that it was kind of the latency factor, because when they got rid of the latency, it was better identifiable, and when it was presumably latent, it was not identifiable. And then also, I think a weak point in the study is that they couldn't isolate the natural killer cells from the mice afterwards to confirm if they had the same behavior of the increase in N-gamma protein, and also the increase, and then also CD107A degranulation that they noticed in vitro when they were kind of comparing whether the CD4 plus infected cells and the allogenic natural killer cells interacted in vitro. Those were the observations they made of the natural killer cells in vitro, but I don't think they were able to confirm that that's how they were behaving in vivo. So they can't really identify the mechanism the natural killer cells are doing to really target these host cells that are harboring HIV, whether active or not. Well, in the active, presumably the active state. And the next discovery I want to talk about is new computational methods for phylogeny. The specific title of the paper is called A Species Level Timeline of Mammal Evolution Integrating Phylogenomic Data by Sandra Alvarez Carretero et al., published on December 22nd on 2021. And what they said is that here we overcome the limitation of previous Bayesian clock dating studies on small data sets by developing a Bayesian sequential subtree approach. So what this is doing is that they're removing uncertainty in time point in which speciation occurred, meaning like we have this new animal species, it like showed up like 2 million years ago or 3 million years ago, like somewhere in between, and they're not really sure when. But now with this new technique, they can pinpoint, like it was like exactly like 1,500 million years ago, and with like 95% certainty. And they can also do this with larger data sets. And so they basically use this with like a geometric Brownian motion method to identify the time points of diversification for over 4,705 mammalian species. So our topic is diversity advancing science. The official title is The Power of Genetic Diversity in Genome-Wide Association Study of Lipids by Sarah E. Graham et al., published on December 9th of 2021. So essentially the argument is that researchers can make better predictions about polygenic effects and functional variants associated with different behaviors with lipids if they included sample sizes that had people of non-European descent. So to prove this, they did a genomic-wide genetic discovery meta-analysis of lipid levels in 1.65 million people, of which 350,000 were of non-European ancestry. Then with small sample sizes, they quantified the benefit of researching non-European ancestries and give evidence to encourage the extension of recruitment of other ancestries, and also show that it's better at identifying loci that are potential candidates that are correlated with these behaviors. So this study is important for shifting away from identifying gene function point of view and switching towards more of a genetic variance for preventative and precision medicine point of view. Okay, we're closing out with a reminder for you to get #boosted. So if you're over 18 years of age, you can get a booster if the minimum time has passed since completing their primary vaccine. So for Pfizer, it's at least 5 months ago. Moderna, at least 5 months ago. Johnson & Johnson, as long as you received your single shot 2 months ago. And you need some resources to get boosted. Well, you can check our Linktree. So if you go to linktree/STEMNewsNexus, you'll see, in addition to our contact information and social media links, this tab that clicks Omicron resources, and it will have links to help you find a place where you can get a booster shot, like right near your house. And And so here's actually one of the links in our presentation that we will actually include in the description of this YouTube video. So thank you. I'm Nina. Have a great day. And and yeah, okay. So yay, science. Woo!